Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic translation. This is from the chapter Molecular Basis of Inheritance. So watch the entire lecture to get basics out of it. So without any delay, let's start with the topic translation. So let's start with the topic translation. So what is translation? It's a process where mRNA is converted to protein. In the last class, I have discussed a transcription, that was the conversion of DNA to mRNA, and the next step after transcription is translation, where we'll convert, or here we'll see mRNA is being converted to protein. So translation is also called protein synthesis. So it is a process of polymerization of amino acid to form a polypeptide based on the sequence of codon in mRNA. So it takes place where in the ribosomes, okay, and ribosomes consist of structural RNAs and about 80 types of proteins, okay, and ribosomes also acts as catalyst, that is 23S rRNA. In bacteria is the enzyme ribozyme for the formation of peptide bond, that is peptidal transferase enzyme in larger subunit of ribosome. So you can see here in the right hand side there is a mRNA, there is a ribosome has two subunit, one is smaller subunit and the larger subunit. And here if translation is done in eukaryotes, it is of 80S ribosome, and if in prokaryotes it's 70S ribosome, okay. So you can see the tRNAs, all right. So here you can see the mRNAs, the ribosome, the tRNA, the amino acids, okay. So we'll see this one by one. So translation, that means protein synthesis, there are three steps, okay. So steps of translation, it's initiation, elongation and termination. Three major steps is initiation, elongation and termination. And before initiation, one more step is there, that is charging of tRNA, okay. So first tRNA will be charged and then followed by initiation, elongation and termination. All right. So first is charging, that is amino acylation of tRNA, okay, that is amino acylation of tRNA. So what is done here, you can see the amino acid, it is in amino acid activating site, you can see the amino acid acceptor site. Then in the presence of enzyme, you can see amino acyl tRNA synthase, okay, it's from amino acyl tRNA. So tRNA will be converted into amino acyl tRNA, that is a charging of tRNA. So again I repeat, when tRNA, so this tRNA have different loops, you can see one side is amino acid activator, amino acid acceptor site, another side is anticodon, then codon, okay. So whenever this tRNA in presence of enzyme amino acyl tRNA synthase, it's converted into amino acyl tRNA, okay. So that is the charging of tRNA, that is amino acylation. So formation of peptide bond needs energy obtained from ATP, and from this amino acids are activated, that is amino acid plus ATP, and linked to their cognate tRNA in presence of amino acyl tRNA synthase. Thus tRNA becomes charged. Next is initiation. So you can see what is initiation. So in this, a small subunit of ribosome binds to mRNA at the start codon, that is AUG. So you have to first remember in the genetic code there are total 64 codon, out of that one start codon and 3 stop codon. So start codon is always AUG and stop codon is UUA, UGA and UAG, okay. So you can see in the ribosome there is a 3 site, you can see P site, E site and A site, okay. P site, E site and A site. So first we'll see initiation. So now large subunit binds to small subunit to form initiation complex. So Smaller subunit of ribosome will be binded with the larger subunit of ribosome. Next, large subunit consists of amino acyl tRNA binding site, that is A site. So A site denotes amino acyl tRNA binding site, and P site represent peptidal site. Okay, so the initiator tRNA which carries methionine, so first amino acid is methionine, so initiator tRNA which carries methionine binds on P site. So first methionine, that is the first amino acid, will bind on the P site. So its anticodon is UAC, recognizes the start codon AUG. So start codon is AUG, so the anticodon has to be reversed, that is UAC. All right, so you can see in the mRNA you can see P site, E site and A site. So first is EPA, so first amino acid will come and bind with the P site. So which one is the first amino acid? Methionine. So which one is the start codon? AUG. And which one is the anticodon? That is UAC. Next is elongation. Okay, so elongation means this process will continue. So the second amino acid tRNA binds to the A site of ribosome. Okay, so its anticodon binds to the second codon on the mRNA, and a peptide bond is formed between first and second amino acid in presence of peptidal transferase. So first amino acid and its tRNA are broken. This tRNA is removed from P site, and second tRNA is from A site is pulled to the P site along with mRNA. This is called translocation. Okay, so second amino acid will enter in the A site. Okay, so these processes are repeated for other codons in mRNA. So during translation, ribosome moves from codon to codon. So by this way you can see a series of amino acid is produced. You can see, okay, proline, glutamine, tryptophan, then glycine, histidine, threonine, methionine. These all are the amino acid, that is a polypeptide chain. Okay, so all the amino acids are joined by a peptide bond. So last is termination. So before moving to termination, these elongation process will continue. Okay, it will continue. You have to remember the first amino acid is P, will be there in the P site and then followed by A. Okay, and this amino acid will move out. Okay, the tRNA will move out from the E site, the exit site, and the chain of amino acid will be produced. So these amino acids are joined by peptide bond and it forms a polypeptide chain, and later on it will form protein. So in the termination you can see when a release factor binds to the stop codon, the translation terminates. The polypeptide and tRNA are released from the ribosome. The ribosome dissociated into large and small subunit. Okay, so you can see a release factor binds. So always you know the stop codon is UAA or UGA or UAG. Okay, so here you could see one stop codon, that is UA, that's where release factor comes and bind, and you can see a polypeptide chain, and all the subunits, smaller, larger, mRNA, all are removed, scattered. Okay, so a group of ribosome associated with single mRNA for translation is called polyribosomes or polysomes. You can see the mRNA strand and numerous ribosomes are there. Okay, so an mRNA has additional sequence that are not translated or untranslated region, or UTR. So UTR are present at both end. Okay, that's 5' end and 3' end. So 5' end is before the start codon and 3' end is after stop codon. So they are required for efficient translation process. Okay, so you can see ribosome larger subunit and smaller subunit. You can see the tRNAs, the mRNAs. So translation in axon, you can see initiation. So you can see the start codon AUG is given here. So in the initiation you can see the methionine tRNA binds to the P site. You can see it's binded. Next you can see in the elongation site the second tRNA binds with the A side. Since P side is filled up, now it will bind with the A side and peptide bond is formed. Next elongation, again you can see the translocation from A side to P side. Okay, from translocation from A side to P side is seen. Next you can see a chain of polypeptide chain is formed. A group of amino acid forms a polypeptide and group of amino acid combine to form a protein structure. Now in the termination, as I have said, release factor binds within the stop codon region. Now you can see all things are dissociated. Okay, all right, so this is how translation process is going on. So major steps before initiation, elongation and termination is a charging of tRNA. Okay, so start charging of tRNA. So these are the major steps. So I've explained the entire PPT or PDF. If any doubt, you can write in the comment section. So these are the steps of translation. Again I repeat: charging of tRNA, initiation, elongation and termination. So before initiation, elongation, termination, first major step is charging of tRNA. Okay, so thank you students. Thank you students. This is all about the entire video lecture. You have discussed about the topic translation. So hope you have understood the video lecture. So thank you students. This is all about today's video lecture, and again we'll meet in the next class. Thank you students.